Hello students, I am back with a new tutorial video on Chapter 3 of plant breeding, focusing on plant introduction and acclimatization. Don't forget to subscribe, like, and share the channel. Let's dive into the tutorial. What does plant introduction mean? How many categories exist, and what is the difference between them? What is the purpose of plant introduction? What are the advantages and disadvantages associated with crop introduction? Our tutorial video today aims to address the questions mentioned above. Therefore, I encourage you to stay with me until the conclusion of this video. What's plant introduction? Plant introduction is taking a genotype or a group of genotypes of plants into new environments. There are numerous reasons for plant introduction among them. The first reason for plant introduction is to obtain an entirely new crop plant. Second, to serve as new varieties. Third, to be used in crop improvement. Fourth, to save the crop from diseases and pests. And the last one is for scientific studies. The other important point in this section is to discuss will be main categories of plant introduction. Generally, there are two major categories of plant introduction. These are 1. Primary introduction. 2. Secondary introduction. And let's what make this plant introduction different from each other. Primary plant introduction. This is when the introduced material is directly used without any modification, or when introduced material is well adapted to the environment. It is released for commercial cultivating without any alteration in the original genotype. However, secondary plant introduction is introduced material may be subjected to select a superior variety, or alternatively may hybridized with local varieties to transfer one or few character from this variety to the local ones. The other important point to discuss in this section is the procedures for plant introduction, or what are the general requirements and procedures you will follow if you want to introduce plant material from abroad to the country. Generally, there are 5 major steps in plant introduction procedure, and let's discuss each of these steps. The first step is procurement, which refers to requesting to an individual or institution abroad, which has the desired germplasm. Then the second step will be quarantine. Quarantine means to keep materials in isolation to prevent the spread of diseases, etc. This step should be strict because it may bring new diseases, insect pests, and weeds to the country. The third step is cataloging, which include giving an entry number and further information regarding name of the species, variety, place of origin, adaptation, and its various characteristics are recorded. The fourth step will be evaluation. This step done to assess the potential of new introductions. For example, evaluation for diseases resistance. The resistance to diseases and pests is evaluated under environments favoring heavy attacks by them. After evaluation of the introduced genotypes, depending on the objectives we have, the superior genotypes will be passed to the last step, which is multiplication and distribution. This step used multiply and distribute the promising introductions after the necessary trials. The other lesson topic we have in Chapter 3 is acclimatization. Let's discuss what acclimatization means and what make it different from adaptation. Acclimatization is a process that leads to the adaptation of a variety to a new environment. It is brought about by a faster multiplication of those genotypes that are better adapted to the new environment. Acclimatization is a quick, temporary change that can be reversed once the previous conditions are met, while adaptation is a gradual, permanent change that is evolutionarily important for a species' survival and continuity. There are also factors which determine the degree of acclimatization. These are 1. The mode of pollination. Cross-pollination leads to far greater gene recombination than self-pollination. As a result, cross-pollination is much more helpful in acclimatization than self-pollination too. The range of genetic variability present in the original population. This means having high diversity equal to high chance of success. 3. The duration of life cycle of the crop. For example, annuals can be selected and commercialized quickly. That's all for today's video. I'll be returning with Chapter 4, focusing on the reproduction system of plants and its relationship to plant breeding methods. Until then, I wish you peace.